what sort of things we want from this system which will allow us to ensure that we've got quality products and processes. So where should we start? Should we start with the people? Why not? So who was doing people? Anybody? Anybody alive? Yes, marvellous. People, right. So, ideas about people. Qualified, right. How do we define qualified? Okay, but how do we define whether they need a degree, for example? Okay, right. So we've got the sort of degree of qualification. But what would define whether or not why would an engineer in a GMP environment need a degree? But how does that relate to the drug product? So the focus has always got to be how whatever we're saying here relates to the process and the drug product. I know what you're saying. So certainly in an oil refinery or somewhere like that, you'd expect an engineer to be responsible for certain things because they would have to sign stuff off. Because it's all about sign-off, it's all about responsibility. From a people point of view in GMP, we have to really go down a lot further to define what qualified means. So really, it's about training. So for each task people are doing, I mean, we can start by thinking about training. For each task people are doing, then they need to be trained for that task. So they need to understand what they're going to be doing. So trained for task. So if we go back to the idea that some people will need a degree because of a level of responsibility, so what do we actually mean by that? So a responsibility of what? Or for what? Okay. So it's their responsibility to determine whether something is quality. So the question is, what happens if that person leaves? What happens if that person's ill one day? Can we no longer produce stuff? So this kind of goes back to the procedures. But one of the key things about GMP, certainly training, yes, training is very important. So people should be trained for the task they're doing. But essentially, they need to be trained for the task they're doing within the constraints of the process. So if you've got someone who is operating the mixer, they need to be trained on the mixer, and they need to be trained to do the mixing step. They are not going to be making decisions. So a key idea in GMP is that the people are not really going to be making decisions. Because when you have people making decisions, they can think yes or no. They can think, is this good or is this not good? And that is a big problem in making a drug product, because we have 3 people operating the mixer on different days. One person may say, well, yeah, this is all right, we mix for 10 minutes, that should be okay. One person says, well, we'll mix for 12 minutes, that should be all right. One person doesn't really care about how long you mix for. So we want to get rid of personal choice. We want to get rid of the ability or the need or even the kind of, we want to get rid of giving responsibility to people for making decisions. So the training is important because people need to be trained in order to do stuff, and we need to make sure that the training has been documented. So the training also has to be documented, and there needs to be a paper trail. But actually getting people to make decisions on the fly, on the go, is generally quite a bad idea, because in cases where people do need to make decisions, again, if there's a decision, it needs to be auditable, it needs to be paper trailed. So I agree, yes, people at various levels of qualification are needed. So some people in this plant will have a degree. Quite often, working on a GMP line, people won't need to be of a terribly high level of education. They will need to be trained well, but it is very repetitive work. It is just operating bits of kit. So the actual need for very highly qualified people working within the environment of GMP will not particularly be terribly important. Granted, people putting this together will probably need to be very highly trained, but highly qualified. But we've got this idea that people actually operating the kit need to know what they're doing, and that is about it. And they need to be able to follow instructions. So, yeah, we've got this training, we've got qualification, we've got documented training, auditable training. So anything else about the people? Anything else at all? So we just stroll into the drug plant and start making stuff. So we just walk in. Hygiene, yep, okay. Right, so cleanliness. And as part of that, we're talking really about dress, aren't we? We're talking about PPE. PPE is the sort of health and safety term, but essentially we're talking about the way in which people are dressed. So this, for PPE is there to protect yourself. The dress that people wear in GMP environments will be to protect the product. So we'll be talking about things, I don't know if you've been around a drug development plant, or if you've been around manufacturing facilities, or if you've been around a food facility. So food facilities have a degree of dress codes. If you go around a food facility, you need to put a hairnet on. If you've got a beard, you need to put a beard net on, which always looks ridiculous. You've got to wear shoes and stuff like that to prevent stuff from being contaminated. So cleanliness and dress is very important in the people in GMP facilities, because they need to make sure they're not going to be shedding stuff into the drug product. So that is rather important. In addition, we've got an idea of health. So if you've ever been and visited a food factory or a drug facility or anything like that, you'll know that when you sign in, you need to fill in a questionnaire, and it says, have you ever had paratyphoid, or have you ever had cholera, or have you ever had, goodness only knows, a big list of diseases. Are you currently ill? Have you got an active cold sore? Have you got the flu? Are you sneezing? Stuff like that. So there's a questionnaire to ensure that people going in not working when they're ill, because if someone sneezes in the drug, then you kind of throw the whole lot away, which is not ideal. Right, so we've got qualified people, we've got trained people, we've got clean people, we've got healthy people doing it, they're dressed in a certain way, that's great. Anything else in terms of people? So if we went to GSK tomorrow and we went to their production facilities up in Scotland, and we wanted to see them making the antibiotics there under GMP facilities, could we? Could we go around the plant? Probably not, no. So only the people involved in the process can be there. So no untrained people. Because essentially you need an audit trail back from the product, back to the product. You need to be able to say, right, this product was made on this date, and at that date, these people were operating the kit, and these people were the only people present. Anything else?
Acting with like integrity and honesty, but facilitating towards like abstract.
Yep, absolutely, yeah, yep. Honesty and integrity. Yep, that's one we don't usually have, but that is very important, because obviously if people are forging stuff, then they'll get chucked out. So how do we actually police that?
Double signatures.
Yep. So we have double signatures. So essentially, an awful lot of GMP is a big part of GMP at the actual production facility will be person presses a button when they're told to press a button by a standard operating procedure. They write down that they pressed the button, they sign that they pressed the button, their supervisor comes over and says, yes, they have pressed the button. And they just carry on like that. So working in a GMP facility is not the most interesting thing in the world, but there we go. Right, so that's people. So what should we move on to next? So let's have a look perhaps at the... What did you lot do down this here? Procedures, right. So tell me something about procedures.
They have to be sterile.
Have to be sterile, right. Okay. And when you say sterile, so what does that mean? What does sterility mean? No bacteria, okay? No contamination. So can we expand that a bit? Can we expand the idea of sterility to encompass more stuff? Clean, yes, absolutely. So sterility is the microbiological way of saying clean. So cleaning is incredibly important. So the big questions surrounding cleaning are how to clean. But even more important than how to clean, I mean, what could be more important than actually how to clean? How clean is it? Absolutely. When is it clean? So is it clean? How do we know it's clean? So do we need to test it? Do we need to validate it? So we need to do validated cleaning. So validated cleaning is an incredibly expensive part of GMP because no matter what we're using, no matter what kit we're using, we need to clean it after we use it, and we need to be able to validate that it is actually clean. Some pieces of kit, so we're looking at a mixer, if we're looking at a crystallization bed, if we're looking at some sort of drying kit, if we're looking at tableting, some of that kit is going to be relatively easy to clean, so an inline mixer will be relatively easy to clean. A sort of packed bed filter will be slightly more difficult to clean. If we're using centrifugation, that's going to be very difficult to clean indeed. So how do we actually validate the clean? Now this will be through sort of two different ways. One is when we have the cleaning, we'll have an SOP for cleaning. And the SOP will need to be based on facts. So the SOP will be, I mean, if we can imagine a very simple SOP for some cleaning we do at home, so if we wash up after we've cooked a meal, so if we wash a plate in the sink, we know what the standard operating procedure there is. You put it in the sink and you scrub it until it's clean, and then you let it dry. So clearly it's an SOP for cleaning pharmaceutical kit is going to be of a similar sort of thing, but obviously a bit more complicated, and it's going to need to be validated. So the SOP will need to be followed in terms of how we're going to clean. There will frequently be clean in place or sterilise in place. So that will simplify things because clean in place and sterilise in place are automated mechanisms by which kit can be cleaned. But it'll be kit that will be bolted onto your manufacturing equipment, and you'll press a button and it will clean it for you, and it'll do it through using a validated procedure. So the standard operating procedure will be designed and developed in order that we can be fairly certain by the end of the process that it's cleaned, and you'll have to do work to develop that standard operating procedure, and HMRA will need to know that that is working and what you've actually done to test the cleaning regime. The second part is after the SOP, you will need some sort of validation. So the SOP will be validated, but in addition, you will need some sort of testing. So that may well be testing that is actually clean, testing whether it's sterile, sterility testing. All of these things are in British Pharmacopoeia. It could well be swabbing, it could well be looking at chemical residues and stuff like that. The other big issue, of course, is frequency. So how frequently do you have to validate your clean? Do you have to validate after every cleaning cycle? Now, clearly, if you've got a big mixer and you're cleaning it and then you validate it, it's not going to be sterile again if you go in it. If you climb into the mixer and take a sample, then it's not going to be clean anymore. So clearly, you can't sterilise it and validate every sterilisation cycle, because every time you validate it, you destroy the sterility. So there needs to be something about frequency, and again, that all needs to be written down into the SOPs. Okay, so we've got something about cleaning. Cleaning is very important. So, anything else around the procedures? Okay, so the materials going in. So grade, supplier, purity, stuff like that. So in terms of this, purity, do we need to test it? How do we validate it? In terms of the supplier, where do we get it from? In terms of the grade, what sort of purity? Many of these things in terms of grade, purity standards are in the BP. So for a lot of materials, the requirements for, let's say, pharmaceutical grade water was in the BP. Same with pharmaceutical grade glucose or excipient or whatever, that will all be in the BP. There are huge tables of what is pharma grade water. The supplier, there will be validated suppliers, and the purity, yes, we may well have to test, but if you buy GMP grade materials, if you buy GMP water, pharma grade water, then it probably will have already been tested. There's always a question in manufacturing whether you purchase something at a lower grade and then purify it and test it yourself to make sure it meets the grade, or you just buy the pharma grade water. And the pharma grade water will be a lot more expensive, but the expense is there because it's already been tested. It'll come with a certificate of conformity, so you know you can just put it into your process and it will be all right, and the FDA or the MHRA will be happy. In addition, how do we actually get them in? Where do we store them? How long do we keep them for? And of course we need the paper trail, so we need to audit. It needs to be auditable. So again, for your box of tablets, you need to be able to audit where absolutely everything that's ever come into contact with that tablet material has actually come from, which is a bit of a nightmare. And of course, all of this needs to be specified by an SOP again. Okay, anything else for procedures? Yep. SOP. Very important. So SOPs for absolutely everything. Right, so middle front. What was your plant? Right. So what do we need to think about in terms of plant? Well, the plant is the equipment.
Yeah. It needs to be adequately designed, commissioned and validated so it's fit for use.
Designed, commissioned, validated. Yep. So, absolutely. Yeah, so for all the bits of kit, the mixer, the crystallizer, the separator, the dryer, the tableter, that all needs to be designed properly. So when we talk about design, what sort of things are we thinking about? What are the important characteristics of a well-designed bit of kit?
It can act in the appropriate ranges that needs to be used.
Okay.
It's safe to use.
Safe, yeah. So it needs to be safe in terms of operator safety, although that rather comes under engineering, health and safety, but obviously it needs to be an important consideration, so that's kind of underlying. The appropriate ranges, yeah, so if you've got a tableter and you're making 1,000 tablets a second, it needs to be able to work in that range. Anything else in terms of design?
Needs to have like adequate HVAC things for it. It depends what the piece of equipment is.
Okay. Consistency, yeah.
Accuracy as well.
Yep. So this needs to be essentially, it's down to the spec, isn't it? It's down to the specification of the kit, so that when you're making tablets, each tablet will be exactly, I don't know, half a gram, plus minus a milligram or whatever. So it's all down to the specification. Is there anything else about the design that relates back to the procedures that we talked about?
It needs to be easy to use.
Easy to use, yep. And I suppose under ease of use, we can sort of say it doesn't want to be confusing. So when I say not confusing, I mean that it needs to be so easy to use that there's never a point at which the operator says, oh, I don't know what I'm doing. I don't know what to do. I will have to make a judgment about this. So it needs to be designed so it is sort of all controllable in a very easy way. Okay. Anything else about the design? Efficient, yep. So the other thing that is usually a very big consideration with design of kit is cleaning. So we talked about cleaning in terms of the procedures, so the cleanliness of the procedures, so no contamination, how do we clean it? An awful lot of that at scale is governed by the design. So it will be very easy to design a mixer which will be impossible to clean. It's a lot harder to design a mixer which is easy to clean. But cleaning of pharma plant is so tricky, and it's such an important step that we have to make sure that it is designed very carefully. So when we talk about design and cleaning, we don't want any dead legs in pipes, so we don't want anywhere for fluids to collect. We want to make sure that when we can clean everything out. There isn't going to be any residues. We don't want corners. So if we have corners that look like that, where stuff can collect, we don't want places for dirt to collect. We want to think about weld and finish. So we want it to be mirror finish, stainless. The stainless needs to be of a specific fineness, of a specific roughness, if you like, to make sure that we're not going to get fouling. So if we can design into the spec of the kit that it's going to be easy to clean and it's not going to get dirty, then that would be a big improvement, because then it means the cleaning is going to be a lot easier. It is an absolute nightmare to try and operate kit that has not been well designed within GMP guidelines. If the kit has been designed well, then effectively it will clean itself. And things like welding, we want to make sure if we have a weld line, the welds are ground down, so it's a smooth finish, so we don't get bubbling or rippling or whatever in terms of the design. So there's a lot to think about in terms of design of plant. We've got the commissioning, and then we've got the validating. I mean, I suppose in the validation, we need to make sure that there is tracking, there is data collection. So for each individual box of tablets, each batch of tablets that come through, we need to think, right, we need to be able to trace this back, who has worked on it, what procedures were used, what raw materials were used, but also, there's a problem with that batch of tablets. Is it caused by a problem here? Can we go back to the readouts from the kit, from the plant, and determine where the problem was? So was there perhaps a problem in mixing? Did it mix for 10 seconds too short? Was the temperature of mixing different? Was the crystallization run not optimal? Essentially, the more we can get that is collected automatically, the more data we can get collected automatically that doesn't rely on a person, the better. Because then we have this massive dossier of data, and we can fairly easily and quickly go back to it and say, yes, this is a problem. And this sort of feeds into preventative measures as well. Because ideally, what we want to be able to do is we want to be able to have our process going through. The plant will be telling us how the process is going, probably in real time. And this will allow us, first of all, to catch any problems early. So if we know that it's got to mix at 70 degrees for 10 minutes, and it's mixing at 72 degrees for 10 minutes, then we might have a problem. Do we junk it? Do we get rid of it? Is it still going to be all right? And again, that will need to be validated, and that will need to go through in terms of the sort of the approved ranges. So in terms of operational space. But in addition, we will be able to then engineer the remaining steps. So if we can still use the mixture that has been mixed for slightly too long or too short, if it's still in the approved range, we could perhaps modify the process as we go along. So there needs to be some sort of tracking and data collection automatically. Okay, right, so in the interest of time, I think, yeah, we can move on from plant. So, guys at the back, middle. Anyone able to speak there at all? What was your topic? Process. Right. Right, so process. So, anything at all? So we've got our process here. So we've got A and B being mixed, crystallized, solid liquid separation, drying the solids, tableting. So is that enough? Is that enough information? Robust, right, okay, so we've got robustness. So the idea of robustness, oops, where are we? Oh, yeah, there we go, right. So the idea of robustness, we need to make sure that the process will perform well at every single stage. So we've got A and B that are being mixed and then going through various stages, and here's the product. So with robustness, we want to make sure that we're generating, if we think about multi-dimensional space at the end of each step, our product is going to be within certain limits, and ideally, our product should be the same at every step. So if we've got 100 kilos of A, 100 kilos of B, we've got a yield from the first step, the second step, the third step, we should be getting a robust process. So we should be getting the same yield in each unit operation. Obviously, the yield will not be exactly the same, partially because there will be variability in each unit operation, partially because you can't be exactly the same because the number will never be exactly the same. But this gives rise to the idea of these sort of ranges of acceptability at the end of each process. So it could well be that you start with 200 grams of material, 200 kilos of material here. We end up with 180 kilos of the product here, plus minus something. What's the acceptable limit? And then 160 and so forth. So this would be our sort of mass. And then obviously we would need to think about other things as well, like purity, like, I don't know, chirality, and various other operational parameters as well. So we've got to have a robust process, and when we talk about robustness, we want to define what we actually mean by robustness. So yeah, so that's good. So what about the rest of the process? You'll notice that it is very vague. So we've got mixing, we've got separation, we've got drying, we've got tableting. So what other information would we need in order to convert this to a GMP process? We'd need some more information, wouldn't we? So we'd need specifications for each of these stages. So we've got mixing. So if we think about mixing, we've got the temperature, we've perhaps got the speed of mixing, the time, pressure, the atmosphere. Is there a solvent involved? So all of these questions, all of these things need to be answered, and they all need to be answered in a way which is specified with robustness, so leading back to the process robustness, but obviously with temperature, is it going to be at 40 degrees, plus minus what? So what are the limits of variability? And this feeds back into the plant. So if we want 40 degrees plus minus 1 degree, that's relatively easy to do. If we need 40 degrees plus minus 0.001 degree, then that's going to be a lot harder, so the temperature control of the plant is going to have to be very different. It's going to have to be a lot more refined and precise. So really, in terms of the process, we need to think about, for each of these stages, GMP needs to encompass what is the actual process, and it needs to be written down in the SOP, again the SOP, with the required level of accuracy. So when we say mixing, we're going to say we're going to mix at 40 degrees plus minus X number of degrees, with the speed is going to be X RPM plus minus whatever. It's going to be for 10 minutes plus minus whatever. We're going to have to think about loading and unloading to make sure that we don't get more mixing of the material that goes into the batch first and comes out later, rather than the last bit of material that you've loaded. So will that still be all right? We need to think about the pressure, the atmosphere, the solvent, all that sort of thing. That all needs to be in the SOP, and there needs to be an indication of how that will all be measured, how that will be validated, and then how that will be checked, and how it has been validated before. So how have we come to the idea that we need 40 degrees plus minus 1 degree? Why isn't it 40 degrees plus minus half a degree? So all of that needs to be validated, and there needs to be justification for why all of that is in place. Okay, so I think we will leave the process there. So guys at the front here, what were you? Product, right. So what do we need to think about the product? Storage, right, okay. So storage really, well, part of that will be distribution practice. But obviously storage in immediately post-manufacture, yeah, that's fine, so there needs to be an SOP for that.
You're going to need to think of an appropriate process for a batch refocusing. If the product's gone out to hospital and there's a problem with it, you can't keep administering it to patients, so you're going to need good batch labelling and an effective recall system.
Yep, okay. So the recall is really under GPP. The batch labelling is a very good point. So each batch needs to be labelled so there's a complete audit trail back to all of the stages of the process. Right, okay. Anything else? Expiration, yep. Okay. And that, again, yeah, that needs to be based on something. So how do we know it expires after a week or a month or a year or whatever? Quality testing, yes. This is usually the thing that people say first. So testing. So how are we going to test? How are we going to test the product? So the big questions there are, I suppose, how and when. So how do we test? We need to test for efficacy, and we need to test for quality, and we need to test for safety. And again, all of this needs to be covered by SOP, all of this needs to be very, very well specified, and all this will contain window values. So if we have an efficacy test, it will be, I don't know, a bio-based test or something like that. Many efficacy tests will be in the BP. For certain drugs, they won't be. It really depends on the drug product. So certain biopharmaceuticals, so anti-cancer medicines, monoclonal antibodies, growth factors, things like that, they have very complex effects on the human body. So therefore, the efficacy test is going to be incredibly complicated. The efficacy test may well take 2 weeks to do because it's a very complicated procedure. Some of them, the efficacy test will be perhaps laid down in the BP, and it may well be a biochemical assay that you can do in 10 minutes. So it really depends upon the product. In terms of safety, yeah, again, the safety testing will be based on what has been used to make the drug. So an example would be if you've got A and B being mixed and crystallised and all of this. If A or B are toxic, then a good safety test would be, is there any of the A or B left? So, and again, you'd need to have some sort of test. Obviously, you can't go through clinical trials again because you're already making the product that's already been approved.
What about if it's the CV drug, like fentanyl or codeine? They need to have some sort of governance involved as well, because if they reach the wrong hands, especially like on the streets.
Oh yeah.
Used for abuse.
Yeah, absolutely. I mean, I think the whole area of drugs which can be misused. So there is a growing problem with misuse of prescription drugs, and as you say, yeah, drugs getting into the wrong hands. Off the top of my head, I don't know whether MHRA governs that. I think.
More the individual trusts and hospitals that
Yeah.
do govern it.
Yeah.
But when they're manufactured as well, because between distributions, they get, things just fall off the back or.
Yeah, I see what you mean. I mean, I think this comes down again to the honesty and integrity of the people. I think because there are so many checks in the process, that it should be impossible for a box of drugs to go missing. It should be. If the whole system is working properly, then a box of drugs will never go missing because there will be a complete audit trail, not only backwards from the box of drugs to the manufacturer, but also forwards from the batch of drugs to where each individual box has gone. My guess, I honestly don't know, but my guess is there are stricter controls in place for drugs which are more commonly misused. That would be my assumption. But I honestly don't know. It's not something it's not something that's been raised in the module before, I have to say. So yeah, it will be an interesting thing to look at. And I would guess the MHRA would be interested in, would then interact with local police to actually control that. So the National Narcotics Agency. Yeah. So in terms of product testing, the other big question is when. So how many do you test? Obviously, you can't test everything you're making because otherwise, if you tested everything, you wouldn't have any products. So you test everything and you just destroy it all. So how much of the product? How many samples per batch? And why? Why have you come to this idea that you only need to test one tablet in 100,000? So what is the validation from that? So every decision you make has to be entirely validated. Right, and then finally, guys at the back, what were you again? Premises, right. So what do we have for premises? Okay, so we have rules for areas in terms of spec, building spec. Yep. Cleanliness. And again, a lot of this comes down to design. So if you're doing the plant design module, you will do this in great detail. So I'm not going to talk about it too much, but essentially, again, just as in plant, the premises, if it's designed properly, then it will work properly. So we need to talk about cleaning. So the design, the cleanliness can be designed in. So we're talking here about surfaces, we're talking about walls, floors, ceilings that can be cleaned. We're talking about the join of the floor and the wall and the ceiling. We've got rounded edges, nowhere for dirt to hide. We're talking about finishes, fixtures and fittings being cleanable. So if you've got a room which needs to be steam cleaned, you can't have light fittings that can't be steam cleaned, you can't have light switches, stuff like that. So it's all about how you can design that in. We're thinking about flow of materials, flow of people. So the clean and dirty streams can't cross. The product stream shouldn't cross back on itself. So A and B should come in at one end of the factory and go in a physical line to the tablets in one direction. It shouldn't cross each other. There should be no point at which we can contaminate or we can mix things from different parts of the process. So we've got the design, and then of course we also have the idea of security. So the idea that that people are going to be dressed in a certain way, people are going to be clean, hygienic. We don't want other people in there, so we need to make sure that the labs are sort of locked down. We can't have other people in the labs at the same time. So I think that's a fairly good sort of summary of sort of the premises. Right, so we've been through the various areas, and I think we've got an idea about GMP. So we've got an idea about the sort of things that are involved in GMP. So what you really need to do is think about and sort of develop from this and develop from external reading the other aspects of GMP. So what sort of things does it encompass? If you look in past papers, you will see that some of the exams in previous years have had questions based on a process flow, and then the question will be, how will this be implemented in GMP? I honestly don't know what's on the exam this year because I've not written it yet, but that sort of question might be on there. I don't know. It is a skill set that is required, though. It's one of the learning outcomes of the module is to have an awareness of GMP. So in this case, it would be a question of how could GMP be implemented and what sort of questions would be asked. Now, I'm not looking for exact answers. I'm not looking for, well, you would have to mix for 10 minutes at 70 degrees and the limits of this would be X and we would have a 10% compliance limit and the window value would be such and such. I'm looking for the next lecture at 11. I will put scans of these onto Canvas, but I should just impress on you that this is not the limit of GMP. This is parts of GMP that we've talked about today, but you need to think about it as well after the session. There are a few more slides on the PowerPoint, but the PowerPoint is available on Canvas. There are a few more slides on that which are on different nations' regulatory authorities, but I'll let you read through those yourself.